host for today. The 3 minute thesis competition or 3MT competition model was developed at the University of Queensland in Australia in 2008. So by 2011, the 3MT competition phenomena had spread around the globe, and now more than 900 universities from more than 85 countries all hold these competitions. And in 2014, UNLV held our very first 3MT, the Rebel Grad Slam, and this year is our 12th annual Research Rumble. The 3MT is a fantastic opportunity for students to learn how to convey who they are and what they study to an audience of folks who are mostly outside of their field. It's an opportunity to build public speaking and communication skills. It's worth noting that our slammers come from all over campus and from a very wide range of discipline. This week, we have over 100 UNLV graduate and professional students assigned to compete in the preliminary rounds. Each will present their research in 3 minutes or less using one slide and no props. We will introduce each student and then start the timer. If a student goes over 3 minutes, they will be eliminated from the competition. Now let's turn our attention to the scoring for this research rumble. The judges will be assessing the competitors based on metrics which total 24 possible points in four major areas: comprehension, content, engagement, and communication. Each judge will independently score each competitor. Judges will not penalize competitors for unanticipated disruptions that are beyond the slammer's control. And at the end of the presentations, we will tally the judges' combined scores for each grad slammer and announce the winners right here live. So the top three in this session will advance to the semifinals. So without further ado, let me introduce the judges, and then we'll go on with a 3MT Research Rumble, which is what we are all here for. We have a set of very distinguished judges in today's sessions. Judges, please feel free to turn your camera on briefly to River Low as I introduce you and share your name with everyone. So first, we have Gerald Geiger from Greenspan Urban Affairs, the School of Social Work. Welcome. Thank you for being here. We have Sean Slattery from Fine Arts. Welcome. Thank you for judging. We have Kim Sharon from the Lee Business School. Welcome. Thank you for being here. We have Jen Nash from Integrated Health Sciences. Welcome. We also have Ashley Blackwell from COLA. Welcome, Ashley. And we have Jen Kent from Integrated Health Sciences as well. Welcome and thank you for being here. We really want to thank you again from serving on our panel of judges today. And now the moment we all have been waiting for is here. We are going to go to our very first presenter for today. Our first presenter is Thomas T.J. Quippen. Who will be presenting on cooling the brain. How low can we go? Thomas, the floor is all yours.
Hello. NASA wants to hibernate humans for deep space travel, and I am investigating the optimal conditions and temperatures that would best facilitate this process. However, we all know that humans don't naturally hibernate, so I began my study by investigating an exceptional example of a hibernator, our friend in the top right corner, the golden mantle ground squirrel. Ground squirrels are unique in that they go through a different form of hibernation than we would typically expect, like of a bear, and instead, they will hibernate for approximately 2 weeks at a time, wake up for a day or two, and repeat the cycle all throughout the winter months. When they go through these hibernation periods, they will reduce their body temperature to the temperature of your average refrigerator, about 36 or 37 degrees Fahrenheit. And furthermore, they will reduce their breathing rate to almost 1 breath per minute. Now, We understand a lot of what's going on with the temperature and metabolism of ground squirrels in hibernation, but there's still a gap in how the brain specifically facilitates these cooling processes without too much harm occurring. So, in my study, I am investigating a cell type called astrocytes. Astrocytes are a star-shaped nerve cell that's found in the brain and spinal cord that facilitates a lot of really important processes in the brain and spinal cord, but ultimately is just responsible for maintaining that nice neutral environment that our brain wants to exist in. So, for my study, I incubated these astrocytes, these ground squirrel astrocytes, for 24 hours at a variety of temperatures to kind of get an idea of what happens when they are subjected to cold stress. On the left-hand side, you'll see that I incubated ground squirrel astrocytes for 24 hours at normal ground squirrel body temperature, about 98 degrees Fahrenheit. And as you can see, they still maintain that star shape. They're interacting with each other. And if you were to Google astrocytes, you would see an image very similar to this. In the middle, you'll see ground squirrel astrocytes I incubated for 24 hours at room temperature, so about 72 degrees Fahrenheit. And on the right-hand side, you'll see the ground squirrel astrocytes incubated at about the temperature of a refrigerator, so again, about 36 to 37 degrees Fahrenheit. And you can see that they start to lose that star shape a little bit, and they seem to almost cluster into these small sphere-like structures, which may be indicative that they're trying to protect themselves. They're in a stressful environment in this cold stress, so they're managing their resources and pulling in order to protect themselves. Now, again, these are ground squirrel astrocytes, and eventually want to do these experiments in human cells, so that way we're able to compare the two and get some sort of idea, like our friend in the bottom right corner, the ground squirrel, just how low can we go? Thank you.
Thank you very much, Thomas. Now we'll be giving our judges 1 minute to score. Thank you. And now we'll be having our next presenter, Joaquin Diaz-Bosignoli, who will be presenting on adapt or die insect decision making in response to extreme cold. Joaquin, the floor is all yours.
Change is the only constant in this world. The environment that organisms live in is always changing, and they must adapt or die. A common example of this is seasonal change, particularly for insects, who have evolved many different ways to avoid dying in the winter cold. In my lab, we study a newly characterized behavior in Drosophila melanogaster, that is, the common fruit fly, who hide away in dark areas to avoid the winter cold. Most likely, this is a way to find shelter against things like winter wind and snow. And this behavior has never before been seen in Drosophila, so the basic causes, like the genes or parts of the fly's brain that trigger this behavior, are currently not discovered, and that's what I'm studying. This is important because if this form of photophobia exists in other insects, and it may very well, then we could use this as a genetic tool to control more dangerous populations of insects, but more on that later. Now, in my study, I place flies in a testing arena which can be rapidly chilled to a low temperature and has a light and dark portion. So the flies have to decide as the temperature falls whether to rest in the light or the dark. And indeed, wild-type flies will hide in the dark spot when the temperature approaches 8 Celsius. Now, this should only occur if the flies can actually tell the difference between light and dark. So for my preliminary study, I compared control flies, marked with the black line on my graph, with flies that were mutated to be completely blind, marked with the red line on the graph. The control flies start out about 70% of the light and go down to only 30% as the temperature drops and they hide away in the dark portion. And the blind flies have no change at all, just as expected. And with this same assay, I can actually figure out the genes that are involved in triggering this behavior. After all, if a gene is connected to this photophobia and we shut down that gene, it should shut down the behavior. And then the fly should choose to rest equally in light or dark. With this method, I'm doing a gene screen looking at a whole suite of genes in Drosophila that are known to respond to cold temperatures to see which one of them is the root cause behind this hiding from the cold. Now, why does all this matter? Well, the genes in insects that control cold sensation and perception are particularly ancient, and they existed across a large amount of insect lineages. So it's very likely that this behavior is evolutionarily conserved. If we could find this in other insects, that would just scientifically be useful to many other entomologists, but more pragmatically, if we can find the similar behavior in insects that are dangerous to human health, like mosquitoes, or insects that destroy agriculture, like the spotted winged fruit fly, then we could potentially use this as a tool to weaken these populations. Imagine knocking out the genes that trigger this behavior to prevent them from finding proper shelter in the winter cold, causing them to die off in that season, and lowering these populations of insects that are so problematic to human health and food production. Alternatively, if we want to support a species of insects, we could maybe try and see if we can sort of magnify this behavior and protect them even further from the winter cold, like for important pollinating insects. Adapt or die. Maybe we can decide for the insects. That's all.
Thank you, Joaquin. Now we'll give the judges a minute to score.
Oh my God.
And our next presenter is Alan Garcia, presenting on After the Final Whistle, Designing the Future of Stadia. Alan, the floor is all yours.
Good afternoon. After the Final Whistle focuses on the issue of aging and abandoned stadiums. While abandoned buildings are not anything new, stadiums represent a unique architectural typology, often costing billions of dollars and holding a unique place in the community's heart and history. Research on adaptive reuse has shown that we have the technology and knowledge to adapt buildings, yet we rarely see it done. In the case of stadiums, cost is often cited as the main limiting factor. But for such a complex building type and a complex problem, that answer is far too simple. This suggests that not all the contributing factors are being properly weighed and valued. Although economics do play a major role, financial concerns don't always hit on all the key factors. This results in abandoned stadiums that cause cities economic and environmental burdens for a very long time. My thesis aims to facilitate the decision-making process to prevent stadiums from sitting vacant for decades and instead transform them into community assets. To do this, I'm going to apply the stakeholder management theory. Through my research, I've been able to identify 10 design criteria that properly weigh and give value to all the stakeholders. This will make the final decision much more actionable and prevent stadiums from sitting abandoned. To test this framework, I'm using Sam Boyd Stadium, a place that we know very well, as a case study. By developing a series of design iterations evaluated through each design criteria to demonstrate why a balanced, community-centered approach is essential. Now, too often we use the word community as a catch-all, feel-good term, but that's not what we're doing here. As part of this research, I'm going to quantify and properly weigh community input, proving that attention to community often results in economic value. This thesis will not only present my own research and my design iterations, it's going to include critiques and contributions from global leaders and local professionals to strengthen its validity. By applying this framework with a focus on community, we can prevent stadiums from remaining abandoned. Ultimately, this thesis will not just provide an answer to what we should do with Sam Boyd Stadium, but it will provide a framework for professionals worldwide to address stadium reuse and potentially serve as a foundation for adaptive reuse of other building types. Thank you.
Thank you, Alan. Now we will give the judges 1 minute to score. And for our judges, we also wanted to mention that Alan had tested his video earlier, so he had a technical difficulty and he was not able to turn his video on, so we just wanted to let you know that. And Alyssa Gardner, the director of the Grad Academy, will be following up with you as well, Alan. And now we have our next presenter, who is going to be Daniel Olgenrout. I am so sorry if I said that wrong. That will be presenting on when software forks apart, smarter ways to share bug fixes and new features. Daniel, the floor is all yours.
Right. We all know Instagram, a tool that we use to share memories and photos. And now we have Threads, another application that has been developed for conversation. Now, what most people don't realize is that Thread actually started as a copy of Instagram. So behind the scene, they share the same code base. Over time, the two applications diverge, and they have different functionalities that exist in different applications. And because of this divergence, it's almost impossible for them to share interesting bug fixes or new features. Now, that might seem small, but imagine that the problem that has been identified, for example, in Instagram, can crash an application or even leave the application vulnerable to hackers. That means if a fix is made in Instagram and the fix cannot be transferred to Thread, so many users will be exposed to vulnerabilities. This is where my study comes in. I developed a tool which is called Repatch, which you can think of as a smart bridge. It identifies interesting fixes from one application and applies it to another application that shares initial common code base. Now, Repatch does not just simply copy and paste the changes, but is able to intelligently understand that the structure of the two applications have changed over time. Therefore, it aligns application, it learns the interesting features that are not common in both applications, and applies them synchronously. This way, the applications remain synchronized and are able to communicate among themselves. Now, the problem that I'm talking about does not just happen in social media application. The same problem can occur in application that we use in our hospitals, in our banks, or even in our cars. If a vulnerability is fixed in one application and cannot be shared in another application, it's like building a bridge on one side while leaving the other side unfinished. My research focused on bridging the gap between the two applications. If application learns to share interesting features among themselves, we have a safe application that can be used across diverse user group. This is where my study focus. Thank you.
Thank you, Daniel. Now we'll give the judges 1 minute to score. Our next presenter is Marlena Gatlin, who will be presenting on retaining administrative faculty, the culture behind commitment.
When we think of higher education, we picture professors, lecture halls, and students. But what about the people behind the scenes, the ones who make sure everything works? What inspires someone to choose that specific path? I'm talking about administrative faculty, if you haven't already guessed. The professionals who coordinate programs and events, recruit students, manage budgets, and make it possible for teaching and learning to thrive. Across Nevada's higher education system, many of these professionals are quietly burning out or stepping away, not because they've lost passion for their work, but because the path forward often feels uncertain. Much of the conversation in higher education centers on student and academic faculty success and retention. But there's another group of professionals whose well-being is just as essential to keeping our institutions strong and running smoothly. Administrative faculty frequently navigate ambiguous job roles, often summarized by the catch-all phrase, "other duties as assigned," alongside limited opportunities for advancement and minimal recognition. These conditions affect more than morale. They jeopardize institutional continuity, equity, and student success. My research explores why administrative faculty, or on the other hand, they leave, and what helps them grow. I'm studying workforce retention and career progression across the Nevada system of higher education. And ultimately, I'm asking, how does organizational culture shape retention and career mobility for administrative faculty? To answer that, I've built my framework around this specific Venn diagram. Each circle represents a major body of theory. The first being organizational cultural theories, led by Sheen's organizational culture theory, which is the foundation that teaches us that culture is more than policies or slogans. It's a shared assumptions about what and who we value. The second and third being retention and motivation theories, explaining why people choose to stay, disengage, or ultimately leave. And the last being career development theories, such as the career construction theories. At the center, this is where all of them really overlap, and it's what I like to call the culture of progression. That sweet spot where employees not only stay, but they thrive in their environment. Sheen ultimately reminds us you can't have true retention without progression, and both are products of culture. That's a guiding idea of my study. Through interviews and document analysis across multiple institutions, I'm uncovering how daily experiences, leadership style, fairness, recognition, and development combine to shape whether people feel they can build a future at their environment. Culture is invisible until we look closer. My study shines a light on the culture that keeps higher education running, asking, what might higher education become if everyone felt seen, valued, and part of the community they serve? Thank you so much.
Thank you. Now I'll give our judges a minute to score. And for our next speaker, we have Monica Carney, who will be presenting on form follows function, the DNA version. The floor is all yours.
Form follows function is a phrase attributed to the popular architect Louis Sullivan, and the premise is that the function of a building will ultimately lead to its shape. For example, if you're looking for a place to live with your family, you may opt for something that looks like a suburban home. However, if you want to, let's say, build the tallest observatory in the United States, you may build something that looks a little bit more like the Strat. Form follows function is a concept that has become widespread and is even used in the study of bacteria. In bacteria, proteins are the functional components of the cells where their form dictates their function. However, there's another component inside bacterial cells called DNA that carry genes or the genetic information necessary for them to survive. Now, it's always thought that this bacterial DNA is unstructured. However, we can see in panel 2 that this DNA is tightly coiled in some areas compared to others, and how tightly coiled this DNA is can actually inhibit the access to that much needed information. Now, there are proteins inside these cells that can control this coiling of the DNA. Some bacteria carry harmful DNA called virulence genes that give them the capacity to make us sick. And unfortunately, those harmful bacteria are becoming more and more resistant to antibiotic treatment, so much so that the CDC estimates that there's just shy of 3 million cases of antibiotic-resistant infections that occur every year right here in the United States. Therefore, it is important for us to understand how these harmful bacteria can turn on and off their virulence genes. I study how virulence genes are turned on and off in the harmful pathogen Shigella. Shigella causes bacillary dysentery, where the infected person will suffer from severe abdominal cramping and bloody diarrhea. What I have done is shown in panel 4, where a protein that is absolutely needed to turn on virulence genes called VirB can actually change the form of the DNA and moreover change the function of the DNA, allowing for virulence genes to be turned on and for disease to occur. If we can reform the DNA, then maybe we can inhibit the function of these harmful virulence genes that allow these bacteria to cause us so much harm. What we're learning is that DNA is not structured without purpose, but rather has many different forms, and those forms serve many different functions in these bacterial cells. Thank you.
Thanks, Monica. Now we'll give our judges a minute to score. next presenter is Haley Strong, presenting on tripping the soundscape using psychedelic to understanding auditory circuitry. The floor is all yours, Haley.
How can psychedelics take us to the summit? The summit I'm talking about isn't the one you see pictured, it's in here. It's understanding how the brain processes sound. But I do believe that as neuroscientists, we can use our understanding of the challenges faced on the summit you see pictured to face our own challenges. The summit pictured is called Mount Eiger, and it sits nestled in the Swiss Alps at over 13,000 feet. And on its north face is a sheer rock wall that has been the fascination of climbers for over 100 years. And that's because of the technical climbing it requires. You must be an expert in climbing on rock, snow, and ice in order to reach the summit. It's a very complex task. And just like this complex task, processing sound is also complex for the brain because sound itself is complex. It's made up of multiple parts. It has pitch, timbre, and it's constantly changing in time and space. And because of that, it's a complex process that the brain does almost seamlessly. But we don't actually understand how the brain is doing this. And because we don't understand how the brain processes sound, we don't have a lot of effective therapeutics for people that have difficulties processing sound. But I truly believe that we can learn from the lessons of Mount Eiger. Because Mount Eiger was only finally first ascended when it was climbed as a team. It took a joint effort of multiple highly skilled climbers who combined their expertise to reach the summit. And I believe we as neuroscientists can join in the expertise of others to finally understand how the brain processes sound. And one of these experts are shamans, because shamans have known for thousands of years that psychedelics are a window into the mind. So why don't we as neuroscientists use them as a window into the brain? If we know that they can change the way we perceive the world, then we should be able to use this to understand the ways that they change the brain to do this in order to understand how the brain processes information. So with the knowledge of shamans and the skill of chemists, a unique psychedelic has been developed that changes the way the brain processes sound, and it changes only sound. All other senses are left untouched. This psychedelic is called DIPT, and it's our window into the auditory world, because now we can use DIPT to understand how the brain processes sound. So just like Mount Eiger, the journey to the top is a team effort, and we've only finally been able to map a route by using a team effort. With the work of shamans, neuroscientists, and chemists, we finally have a map to the top, and all that's left is to climb. Thank you.
Thanks, Haley. Now we'll give our judges a minute to score. And our next competitor is going to be Ashley Godfrey, speaking on When Crowds Crush, Lessons from the Lived Experience. Ashley, the floor is all yours.
I remember the exact moment that my feet left the ground. I was in the middle of the crowd, just like this this one, and the crowd surged around me. I was being carried by a wave of people that I couldn't control. At another concert, the crowd was packed really tight and hot, just like this one, and I passed out. The next thing I remember is I was waking up outside the venue with the paramedics. After both of these times, I was able to walk away, but those experiences never left me. That's why I'm studying crowd crush. Since the year 2000, over 4,300 people worldwide have been killed in crowd crush incidents. And I'm looking to help prevent these disasters before they start. And what's most alarming is that we don't fully understand how or why it happens. My research explores people experience and manage crowds at live music events. We are looking to, again, prevent the disaster before it happens. So I have several ongoing research projects that I'm working on to look into this. First, I'm doing a literature review to uncover what we know about crowd crush. This is an under-researched area within hospitality and tourism. Most research focuses on crowd density models, but not what it actually feels like to be in the crowd, like in this photo. Second, I'm doing qualitative crowd observations at live music events where I'm looking at how crowds move and how they develop risk areas like at barricades and at bottlenecks. Third, I'm doing crowd interviews with concert goers, just like at the front of this photo here, to understand more about their lived experiences and what makes them stay in unsafe spaces and what safety means to them. Finally, I am doing a survey, quantitative, for event managers that focuses on how likely they are to adopt new safety technologies that would work with the barricades, like you can see in this photo, a smart barrier technology that can help detect dangerous levels of crowd pressure before it's too late. This research matters because every person in this photo deserves to go home safely after an event. And in today's world, millions of people come together every year for live music events, and it's not optional to understand crowd safety, it's essential. My research looks to bridge the gap between crowd experience and crowd safety to bring together the human experience and the data. Thank you.
Thanks, Ashley. Now we'll give our judges a minute to score. And our next speaker is Salome Ogbuleka, who will be speaking on Built for Access, how neighborhood design is associated with workability and food insecurity across U.S. communities. Salome, the floor is all yours.
Thank you. Remember the last time your car broke down or was parked at a shop and you probably need to get around your neighborhood to get something, which necessitated you to walk, maybe along a stretch of a road in the scorching heat of the sun with no shade and cracked sidewalk. It probably felt inconvenient, maybe even unsafe. Now, imagine doing that every day to get food, medicine, or other essentials. For many low-income families, that's not a bad day. That's everyday life. Food insecurity is already a heavy burden. Families are unsure if they will have have enough for the week. But what if on top of that, your neighborhood makes it nearly impossible to reach the nearest grocery store? Think about families without car or older adults who once work but now avoid it because it feels unsafe and scary. To them, it's not just about the food, but how to get the food in the first place. To explore this, I use the geographic information system to map patterns across neighborhoods. I use datasets from the National Workability Index that measures how workable a community is and food insecurity data from CDC places. By layering this dataset on the map, I can see where low workability overlaps with high food insecurity, where people are most at risk. Here on our slide, we can see a special distribution of workability and food insecurity in Nevada and the Las Vegas metro. On the Las Vegas metro map, we can clearly see the different colors on the map, which shows the different levels of workability, from least workable, below average, above average, and most workable community. Then the different shapes on the Las Vegas metro map, colored blue, orange, red, sky blue, shows the rate of food insecurity as dispersed in this area. Why does this matter? Food and the built environment are social determinants of health that shape how people live, move, or access daily essentials. And so, overlooking the built environment means overlooking a critical piece of the food security puzzle. This research will help city planners, design professionals, and public health officials to design a more connected, equitable, and food secure community. And so when I ask, are our communities truly built for access? I am not just talking about the aesthetics of architectural or engineering roadway design. I am asking that we build a more just and resilient community. Thank you.
Thank you. And now we will give our judges a minute to score. And our next speaker is Gabson Baguma, who will be presenting on From Gray Water to Lifeline. And don't forget to turn off your camera, Salome. The floor is all yours, Gabson.
Every single day, an average person uses around 100 gallons of clean water, and behold, almost 60% of it is sent down our drains as gray water. So now imagine if this just reused water can be saved enough every year to meet the needs of over 1 billion people living in water-stressed regions. So that's the scale of waste and opportunity that is hidden in our homes. In communities which are far much stricken in desert areas, for example, the case of Nevada and the place where I come from, like Uganda, in rural areas. So the water scarcity is not just a problem, it's not just a statistic, it's their daily struggles. And in this, families usually compete for the limited supplies, the utilities which are rising from treated costs, and extremes worsen in these cases. Yet, in most of this water reuse or bathing or cleaning or laundry, it can still be clean enough and can be reused if only we had the right technology. So my solution is, my research asks this simple, radical question. And it would be, what if this wastewater or the gray water which we, lose down our sinks could be turned into the most part of the solution for this water scarcity problem? So what we are doing is we are developing a solar-powered gray water filtration system, which is built on readily available materials, which are locally available to us, the scrap iron, the sand, and the biochar that can convert or remove these contaminants in the water we release just down to the sink, and which is from our kitchens, which is from our bathrooms, and we can recycle it for non-safe drinking uses, for example, cleaning or gardening and also sanitation. So why does, in this case, why does this one matter? So this is not just about engineering in this case. It is more of the dignity or the recycling of the water. So if we saved this water, it's probably expected that about 35% or 35 gallons a day could reduce the demand on the scarcity of water, and then we can also cut the pollution levels and can give these rural communities control over their water. So water scarcity is not just about fancy things. It's just humanity's greatest challenges. But there's a solution that can be done that is just flowing beneath our sinks. So if we change how we see this wastewater not as just water but untapped opportunities, then we can drop the reuse, and save more of our water. So it's not just about research here, it's about the saving communities, and it's just a movement to turn every drop into a lifeline in this case. Thank you.
Thank you, Gabson. And this was our last slammer for today. So we thank all of our slammers and want to thank everyone for joining us for our preliminary rounds. Thank you to the Graduate College team who have made the 3MT event possible. Now just stay tuned. We'll announce the winners live here in just a few minutes. Judges, please be sure to press submit at the bottom of your scoring rubric form so that we can get to see the scores. While only the top winners of this round will progress to the semifinals, you will all be winners of a Rebel Grad Slam t-shirt. So you can get yours during the Rebel Grad Slam Finals reception that we hope that you will all be there for that. So for the preliminary rounds, the first place winner will receive a $300 scholarship. The second place winner will receive a $200. And the third place winner will receive $100. So in just a few minutes, we will have the names, we'll announce the winners. In the meantime, I just wanted to thank you again, and don't forget that the semifinals will be on Thursday, November 6th from at 9 a.m., 11 a.m., and 1:30 p.m. in the Greenspun Auditorium. And the finals will be on Friday, November 7th at 4 p.m. in the Greenspun Auditorium as well. We'll welcome you to RSVP for both of these events. And now we'll just wait for a few more minutes. I know we are all excited to know who won the rounds, but you all did such a beautiful job. And I can't wait to see who I'm going to see in the semifinals. But you all did such a beautiful job presenting the amazing research that you are doing here at UNLV. And we are all so proud of you here at the Graduate College. Well, hello everyone. Welcome back. We have our winners, and I'm excited to announce them. And I am going to start with third place, then I'm gonna go to second place, and I am going to leave the first place for last because we have a four-way tie. We have four people at the first place, which has never happened before. And this is happening in my session. How exciting is this? I feel like the most luckiest host that this is happening at my session. If it would happen at any session, it would happen at my session. So I am so excited. I am so excited to share this. So we are going to start. So for our third, you can give me like a silent drum roll. So our third place is going to be Marlena Gatlin. So you won third place. And for yes, I see a lot of claps going on. And for our second place, we have Salome Ogbuleka. And second place, congratulations. And now for our very first place, we are going to be having four people is advancing. We have our four first place. So the first one is going to be Thomas Griffin in first place. And we are also going to have Joaquin Diaz Wosignoli in first place as well. We are going to be having Monica Carney in first place as well. And we are going to have Haley Strong in first place as well. So, congratulations to all of you. Thank you again to everyone who competed. Thank you to all of our judges. Thank you to everyone who made this event possible, and we can't wait to see you at the semifinals round on Thursday, November 6th at 9 a.m., 11 a.m., and 1:30 p.m. in the Greenspan Auditorium. And the finals will be on Friday, November 7th at 4 p.m., also in the Greenspan Auditorium. And lastly, we will have your Rebel Grad Slam t-shirts available at the final reception in the lobby for everyone who actually presented today. We welcome you to RSVP for the finals event, which the link should be provided in the chat for you. If you did not RSVP yet, please go ahead and RSVP. It was my pleasure to be your host for today. Have a great day, everyone. Thanks for coming.
Thank you.
Really quick, when do we find out the times of the semifinals?
Yes,
Oh,
we
yeah.
will be, sorry, yes, the Grad Academy will be sharing that out by the end of the day, Wednesday at the latest, as we wait for all the rounds, but we'll try to share as soon as possible after 12 p.m. on Wednesday.
Okay. Sweet.
Thank you. Congrats, everyone.